Hi everyone, my name is Shin Yi. Today, we are addressing a critical issue, oil spills and fuel contamination. These issues have far-reaching implications for our environment and food supply. In the next 10 minutes, my team will explore a solution, bioremediation. Bioremediation, the use of living organisms to remove pollutants, is not a new concept. It dates back to studies on oil and sewage treatment as early as 1968. The first large-scale bioremediation effort was in 1972, addressing the Sun Oil pipeline spill. Today, we are taking a novel approach. Scientists are isolating microbes from car fuel tanks for bioremediation. The reason? These microbes living on fuel sources like gasoline or diesel have the potential to clean up spills. The convenience of the fuel tank environment also makes it an ideal source for bioremediants.
Hence, studying car fuel tank microbiomes can allow us to potentially isolate hydrocarbon-degrading bacteria, which can be applied to rescuing soil and groundwater contamination caused by fuel leaks and spills. Before we begin, here are some terms to enrich your grasp of our sharing. Bioprospecting explores natural bioresources for medicine, agriculture, or industry use. Next, biodegradation is the breakdown of organic substances by microbes, making them more environmentally friendly. Lastly, biosurfactants, nature's dishwashing soap, reduces surface tension and enhances liquid dispersion. Now Adi will be talking about the study aims and design.
In this study, the researchers aim to study the taxonomic profile of the microbes under the car tank lid and specifically their potential in biodegrading field. The second study aim is to identify the strains with the highest potential to biodegrade diesel. As for study design, the study starts with sampling by collecting dust under the tank lid or filling point. The biomass collected will later sequenced with 16S rRNA gene sequencing. Some of the biomass collected was cultured with either gasoline or diesel. 16S rRNA gene sequencing was performed on the fuel culture. From every fuel culture tube, the pure cultures were identified and collected. The 16S rRNA of isolated pure culture was sequenced. The pure culture collected will be tested for bioemulsifying potential. The strains with the best emulsifier production were selected to undergo fuel degradation assay. 16S rRNA gene sequencing were done on all three cultures to study their taxonomic profile, and the screening part was done to identify the best strains with the ability to biodegrade diesel. Next, Rhino will continue for the result.
As mentioned previously, bioprospecting was performed on the 10-lead bacterium using extensive sequencing, selection, and screening. The initial sequencing of 10-lead biomass uncovered similar bacteriums in gasoline and diesel tank lids. However, they still displayed variations when compared to bacteriums in other hydrocarbon polluted sources. These similarities is evident from the box plots, which portray similar richness and alpha diversity between bacteriums of both fuel sources, with no significant difference in beta diversity. Particularly, both bacteriums share a resemblance in their taxonomic profiles, dominated by proteobacteria, acinobacteria, and cyanobacteria at the phylum level, with 10 highly abundant genera. These bacteriums, when contrasted with those from other hydrocarbon polluted sources, demonstrated a much lower alpha diversity, with distinct variations in microbial communities, underlying the unique niche of fuel tank lids. Moving on to the analysis of bacteriums from gasoline and diesel selective growth cultures, the authors stumbled upon a reduction in diesel microbial community compared to the initial biomass. Only 3 genera, dominated by Pseudomonas, were identified. Interestingly, these genera were undetectable in the initial biomass samples. When bacterial strains from these fuel cultures were isolated and sequenced, a larger number of culturable strains and genera were identified. A distinct set of bacterial strains and genera was found in diesel and gasoline cultures, as depicted in the diagram, with 38 culturable strains in diesel compared to 9 in gasoline. Notably, only 2 culturable genera, Bacillus and Isopatricula, were observed in both types of pure cultures. Regarding the functional capacities of these bacterial strains in bioremediation, the authors noted a high proportion of strains that could effectively emulsify gasoline. In contrast, only a few strains exhibited effective emulsification of diesel. With these results, they selected the 5 best strains for fuel degradation screening. The screening highlighted the variations in diesel degradation capacities of selected strains. All strains displayed the ability to degrade small hydrocarbons, as indicated in both diagrams, with no black peaks observed at low acquisition time, indicating the absence of small hydrocarbons in degraded fuel, compared to the blue controls. However, only a few strains could degrade larger hydrocarbons. These strains expectedly exhibited a higher total diesel degradation between 50 to 60%. Now, I'll pass it out to Ingrid to discuss the implications of the results.
After obtaining their results, the scientists sought to explain the phenomena they observed. First, they reasoned that the diversity of the microbiome samples was driven by their ecological niche rather than fuel type or carbon source, as the diversities of the car tank lid microbiomes were slightly lower than the diversities of both the polluted soil and the solar panel microbiomes, regardless of the type of fuel used. In addition, they hypothesized that the similarities in composition of the tank lid and solar panel microbiomes could be due to the different selection pressures microbes face on artificial and natural surfaces. Microbes on artificial surfaces experience higher temperatures, dryness, and a lack of nutrients as compared to soil. Furthermore, the car tank lid microbiomes are confronted with an especially low nutrient environment that is rich in hydrocarbons, potentially explaining why the tank lid microbiomes were less diverse than the solar panel and soil samples. However, this hydrocarbon-rich environment is also what gives car tank lids the potential to harbor hydrocarbon-degrading microbes. After growing the car tank lid communities in enriched cultures of 10% gasoline or diesel as the sole carbon source, the genus Pseudomonas, a well-known alkane and hydrocarbon degrader, became the most abundant genus in diesel cultures. The scientists also stated that it was remarkable how it was not detected in the initial microbial samples, suggesting that it was present at only a very low abundance on the tank lids. Biosurfactants help in degrading hydrocarbons by emulsifying them. Bacteria from genera such as Pseudomonas, Bacillus, Achromobacter, and Isotaricola were shown to be biosurfactant producers, and the enriched fuel media increased their biosurfactant production. Lastly, the researchers discovered a new Isotaricola strain, and sequencing its genome revealed that it synthesizes several enzymes involved in various hydrocarbon degradation pathways. In short, car tank lids are micro-niches for microbes which differ from the polluted soils traditionally used to isolate hydrocarbon degraders, and they even contain bacteria that could be used for fuel biodegradation. Next, Marie Lynn will be addressing some of the limitations of this study and the future work that can be conducted.
Despite the researchers addressing most of their aims and objectives, there are still some limitations in the study and opportunities for future work to be done. For example, the sample size of 19 is quite small, with one sample being discarded due to low amounts of DNA. Future studies should consider low microbial or DNA load during sample collection and increase the sample size to account for this. Additionally, the microbes found in the sampling area of the car tank differ depending on where the car has driven, thus the results may not be representative of vehicles found outside of Spain. Another recurring limitation in the study involves the exclusion of gasoline samples from sequencing and fuel degradation experiments. This is because gasoline evaporates faster than diesel, so it is harder to quantify it. While this may not drastically impact the study's conclusions, it does reduce the comparability between the two fuel sources through selection bias. The authors also suggested that a new hydrocarbon degrading strain was identified during the study. However, more research is needed to completely describe and outline the degradation pathways that take place with this new strain. By exposing this new strain to environments that simulate oil spills, the author could potentially better translate the study's findings to the broader purpose of bioremediation. Lastly, it is important to remember that microbial communities, rather than individuals, are involved in fuel degradation. Thus, future studies can be conducted on combining microbial communities with different enzymatic or emulsification properties to assess their effects on different hydrocarbons. Additionally, sampling microbes from other parts of the car, like the engine, may provide insights into useful microbes, such as thermophilic bacteria, for other applications. With that, we've come to the end of our presentation. Thank you for listening.